All right, I think we will go ahead and get started. Good morning, everybody. My name is Tori Colburn. I'm the Midwest Conservation Agronomist at American Farmland Trust, and I'm the co-host of these ICON calls. Please, I would like to welcome everyone to our March 13th call in the series of soil health testing. Today, we are fortunate to be joined by the director of the Cornell Soil Health Laboratory, Robert Schindelbeck. Looking forward to hearing his presentation today. Before we get started, I think as usual, what we want to do is have everyone put their name, their organization, occupation, and your location in the chat, just so we kind of know who everybody is and who's joining us for the call. And I think we'll go ahead and open our entry poll too. While we're doing the poll, just want to make sure everybody is aware that we are going to be recording these calls, and they will be posted to our YouTube page. So if you have to duck out early, or if there's somebody in your organization who has unable to join us that wanted to, they can view the presentation later. And then do we have, I don't know if I can't see our poll results, so you'll have to tell me when we're finished with that. There we go. Okay, hey, well, we have folks who are familiar with some soil health testing. That's good to see. Hopefully we'll expand our knowledge today. Okay. Okay, so just to go through the quick rundown of what today's call is gonna look like, we're gonna go ahead and kick things over to Bob Schindelbeck for his presentation, and then we'll have an open group question and answer session, and then we'll wrap things up with some updates from ICON and ISAP, as well as going over what our upcoming calls will be, and then we will open it up to the floor for any other organizational updates that you all might have. So with that, I think we're ready to kick things over to Bob. So, Bob, if you want to go ahead, I think we can go ahead and I guess I'll introduce Bob a little bit more. Bob is the director of the Cornell Soil Health Laboratory at the Department of Soil and Crop Sciences at Cornell University. He's going to be presenting today on the Cornell Soil Health Assessment Test. So we'll go ahead and kick things over to Bob. Bob, I'll let you take it away.
Thanks, Tori. Okay, I've just enabled screen sharing. I'm a little slow here.
Yep, we're seeing your screen, and you can go ahead and go into presenter mode. There you go.
Great. Okay, well, good morning, folks. I'm going to talk today about soil health, as you heard, as you understand. A little bit of background about myself. I'm the director of the Soil Health Lab at Cornell. Cornell University is the land-grant university in New York, and believe it or not, New York State has huge amounts of agriculture. People think of New York City. That's not just it. I mean, New York is. I think New York is like #1 in sweet corn. We're in the top 2 or 3 in potatoes, top 2 or 3 in carrots, maybe even cabbage. We're up there in maple syrup. You just really wouldn't believe all the different crops and commodities. Long Island, it's kind of, it seems like a suburb of New York City. Huge. There's this huge sand, just a sand deposit stretching out into the ocean that gives Long Island a climate about like the Carolinas. And it's just a huge area, just so much agriculture out there as well as people. But it's really a surprising place. So we've been involved in soil testing and just agronomics and vegetable production in New York for a long time. And I can recall it was actually 2002. I was trying to say 22 years ago. We started talking about soil health. I heard it for the first time. We used to call what we call now soil health as we'd call it sick soil syndrome. We'd look out at a field and we'd test it. And I'm showing 3 spheres here on the left, 3 spheres of color, showing these spheres kind of balance. That's the 3-legged stool, kind of balanced with soil health in the middle is the idea there. And if we would measure back then, we'd get our soil chemical results, but the field would look terrible. The plants would look bad. Things weren't growing well. what's going on, and we could guess, we could surmise, I guess. But there were no tests. There's no test to kind of test these different parameters like soil physical structure or water holding. There were research ways to do that, but for the public, there wasn't a way to kind of get that information. So that's where, for us, the soil health testing was born out of that. We wanted this extra information. And so what we did, here. Right, we wanted to put numbers on some of these other parameters besides just the soil chemistry. So I'm going to start here on the right, talk about these three spheres again, the soil chemical testing. I've got little pictures here to go with the chemical ideas. That's the meat and potatoes picture. Nutrients are fundamental to soil testing. We've been doing it a long time. We know an awful lot about levels and rates and amounts that we need, proper pHs for different crop plants. So we want to kind of build on that. And then if we could move to the bottom, the biological testing, the soil is alive. It's a living entity, and it relies on carbon as the energy currency. And so there's all these different ways to measure carbon. And so we look at the amounts of carbon available for energy to feed these different organisms. You'll see it in a minute with active carbon. We look at the organic matter decomposition rates, and we'll talk about how, why would we want to decompose our organic matter? Aren't we trying to build it? That's kind of a paradox, but that's what the health is all about. Nitrogen supply from that organic material, and of course, nutrient cycling. All that organic material was once alive. Any of that organic debris was once a functioning root or stalk or a leaf. They had a full complement of all the essential elements. And so now it's laying there in this different form, but it's kind of got everything in it. It's got copper and boron and everything else, sulfur, that could still be available for the soil food web to break down and make available to plants again. And of course, the soil physical environment, I draw that as a house. That's the kind of the place where certainly the roots live, and the plant is anchored there. It can't leave. It's kind of stuck there. And if it's not a good physical environment, you can't take advantage of all the nutrients that might be there if the soil is compacted. The roots can't get to that nutrition. Same with the biology. The biologicals are releasing all sorts of wonderful compounds for the plant to use, but if it can't get it, if the soil is too dry and hard, it's just not going to happen. So we need that balance. So the physical, I'm going to go through these. I like this one. The structural stability, that's what we call aggregate stability, or the crumb structure. It's the structure of the soil. You need a good structure to hold up against erosion, of course, but also to intake water. So a soil that seals over can't take in that water. Of course, that means you're going to be droughty later because you didn't get that water in now that the rain came. And also, as the water moves in, it softens the soil. And not only stores it for later, it softens it. Of course, as the water moves in a structured soil, as it passes through the soil behind it comes fresh air. And so it's this physical structure is just so important. I spent a lot of time thinking about this myself. It's just the place where so much happens, where everything has to take place in. So if this is upset and compacted, for example, the roots just can't take advantage of what all the good things that are happening down there. Okay, so going to talk about the soil health testing lab. This is where we do the soil health test report. You see this on the right. We opened the lab in 2007, so it's almost 15 years. It is 15 years. They were doing this general test with all these different parameters. The big idea is there's all these different. Well, you see the three groups. First of all, the three colors, and of course on the report. Use my mouse here. There's the physical, biological, and chemical tests that we perform in the lab. And these are essential soil processes. And these are things that just we know that are important in the soil. These are things that we can measure kind of quick and dirty. They're reliable. We kind of know what they are. They're easy to learn about. They really tell us something with that information. And so we're gonna measure those. We'll go over those real quick. You should know this whole test, this whole suite is $130 per sample. It's a lot of money. But it's a whole lot of information. And what I really want to talk about is how why all that information is important and how we can get the most out of it so that we're not spending a lot of money and then only using a fraction of it. What you see here is the first page of the report, the summary first page that, of course, lists all the tests here. There's a lab value, there's a rating, color-coded rating, and finally constraints. So this first page kind of just, it summarizes everything. The next pages all talk about what are these tests, just quick and dirty, kind of going into what your value is, what the score is, what that means you're in the red zone. That's not so good, how they're constraining the soil. We'll get to that, but this is the first page. And these are the tests. I wanted to just show these real quick. I stuck bulk density in here because that's another test people are asking for more and more. I certainly want to go with carbon testing to get total carbon per acre. But we do a soil texture test. This is in our standard package. We do a soil texture test. There's a soil hardness component using a soil penetrometer. Of course, we can't do that. That's information you have to provide on the soil submission form. It's optional, but we loan out penetrometers. We mail them all over. There's we got a fleet of them that are out. And people can take the readings at the 0 to 6 inch depth and 6 to 18 inch depth. They just stab this thing in the ground in different places, record the highest number on our submission form encountered in the 0 to 6 and 6 to 18 inch depth, get an idea of how hard your soil is, and we can score that and feed that back to you on this report. So all that information is in one place. Water storage capacity. I'm showing a pressure plate here. Then our aggregate stability test, that's a soil structure test. You can see the little raindrops coming down on soil samples that are on these sieves, full-size sieves. It kind of shows our rainfall simulator on the far right here, how we do the test. We rain half an inch of rain in 5 minutes. We put a thunderstorm on these crumbs and see how many of them fall through this sieve and how many are retained. That gives us an idea of how stable the soil is, how tough it is, how it holds up against the environment. And then the biological parameters, we've got this active carbon here. That's that permanganate oxidizable carbon. A lot of you have heard of that one. We do a 4-day soil respiration. This is an index of the amount of breathing that goes on in the soil. Of course, that means carbon dioxide release. So that means carbon is being used. But we want this number to be high. It's an idea of the idea of right, if you will, an athlete's muscles use more oxygen than the fat tissue on the person who's not an athlete, let's say. And so we want to measure that. And if we can measure more respiration, we might say there's more muscle in that sample, and muscle is a good thing. Then we go on to soil protein. This is an index of soil organic nitrogen. Proteinaceous compounds have nitrogen in them, sulfur, organic. And that, of course, that would contain, that would be, that's DNA, RNA, cell walls have protein in them, little parts of insect wings, little parts of plants, part of a seed of a soybean that's left in the field, that would be measured in the soil protein. But again, that's an index of the soil organic nitrogen. Then we do another test, total carbon, total nitrogen. And then we also determine the organic carbon content, which is the one that matters. So sometimes total carbon is quite high, but it's organic carbon that counts, particularly if we have a lot of lime in the soil. We get a lot of carbonate. Remember, think of calcium carbonate being lime. That would give me a lot of total carbon in my soil, but that's not what, if I'm doing this for subsidy payments, no one's going to pay me for my inorganic carbon, the carbonate. They're going to pay me for organic carbon. So that's why we include the organic. And of course, we also do this standard old-fashioned organic matter percent, where we burn a sample in the oven to get the loss on ignition. And of course, the chemical nutrients across the bottom. Up here, we use a modified Morgan extraction. That's weaker than certainly Malic, but 10 times weaker. Soils up here are very young, just 10,000 years old. So we need just this weak extraction compound. This is what we use. But we then score. I'll show you how we score that later. But then, of course, we do pH 2. But this is just the technique we use. Then soil digestion. This is for heavy metals. This is a microwave machine, and where we put samples in these little tubes here, Teflon tubes with nitric acid, and we boil the samples in boiling nitric acid to totally digest it, so we can get the heavy metals out. And then, of course, all this is measured on the ICP for each element. Okay, back to our soil health report, this first page. Going to go over to the report. You see this black arrow here. So here's the value from the lab for each of these different parameters that we measure. And of course, then on page 2, we describe how we get this rating. So each of these parameters gets a rating. There's a color-coded score. It's very important. Things like, well, if I did a human health test and my serotonin came back at 280, I'd say yes, and I'd want to know, is it high? Is it low? At 280 doesn't help me. I need to know what range that is. So this rating is so important. And we rate everything based on texture. We just break things into 3 general texture classes. Fine, those are clays. Mediums, silt loams would be medium texture. And coarse, the sandier textures. And then we take this number and say, for a sand, or in this case, for a silt, is that good or bad, or indifferent? And you see this color right here for available water capacity, 16%. What's that rating? 52. It's in the yellow zone. So there's actually a bell-shaped curve here of all our thousands and thousands of samples in our database. But the idea is, if you're in this yellow, you're kind of in the middle of all the other silt loam soils for this parameter. If you're in the red zone, like the surface hardness for this sample, that's 260. That comes back at 12% as a rating, and that's down in the bottom 20%. So that flags as red. That's something that you could probably, that should get your attention, something you think about. Why is that number like that? And so we pop out this constraint. We'll talk about this in a minute. That's affecting the soil's performance with respect to plants. So that soil's going to, the surface hardness, it's too hard. That's going to affect your rooting, water intake, water transmission, all these issues. Something to think about going forward. Of course, here's the same sample. This is quite a. I'm going to show you this soil in just the next slide. But first, I want to show you that there's quite a few things going on here. First, down at the bottom, below this bottom heavy black line here is the soil chemistry. Looks like everything's hunky-dory. If all I did was a soil chemical test on this sample, I'd say, yep, good to go. Get out of here. Go grow your corn. But of course, we can see, if we look at these other things, and you might see this by looking at it in a moment, there's other things going on. There's other parameters that are out of range that are going to hold you back. So this is not unlike if you weren't feeling well, you'd say to the doc, I'm just not well. What's going on? If they just did blood work, they might not catch certain things. But if they tested other parameters, liver function, your strength, that's not blood work. If they did some sort of a test where they have you resist them pushing on your hand or your arm, just did a more varied suite of tests like we have here, they might find something that makes you think, oh yeah, that's something we should look at. That's a way into something that's out of range, particularly if your blood work, meaning the soil chemistry, all looks okay. Of course, this is showing here on the summary first page. We also have an overall quality score just to bring everything together. But generally, we're always looking at each of these individual things to kind of understand what's going on individually. Oh, let me go. I want to go back. I'm sorry. Yeah, so I've highlighted an extra pink here, this aggregate stability parameter. So this is that soil structure test that we do. If you notice the value is 13%, and that's in the red zone. Not good. That means we're probably going to have an issue. So says my text here. It's going to have a problem with aeration. The soil's going to seal over with infiltration. Rooting, soils will be able to root. The soil's too hard. Going to get crusting, sealing, runoff. There's going to be erosion. All these sort of issues. This is what the text says. But on the left here, this is that soil. The soil sample I just showed. This is what it looks like in the field. A over here on the left. And that same soil, this is a columnar silt loam, a fairly common soil around here, certainly around campus. Under intense cultivation, the plant breeders use this field every year for growing their wheat to keep their their seed stocks up. So they beat the heck out of it. That's okay. It gives us a chance to measure a beat up soil. And on the right, it's the same dirt about 100 feet away. So we flipped it over, the sod outside where they grow here. And this is what this soil looks like if you didn't intensively cultivate it and use it hard, of course, under ideal management. So we put it through our test for the aggregate stability. And on the left, you see what the sieve looks like. This sieve was loaded with one layer of soil crumbs, about 30 grams, on this 8-inch sieve. And about 13% of it is stable after we perform this test. And on the plowed sod, about 53% is stable. That's actually good. I'll show that. So now I'm showing on the left, this is the report we've seen before. Of course, this is soil A on the left. And you can see it's got those issues. Again, extra highlighted the aggregate stability here down in the red zone. But I think if you look at this soil, at soil A here, you would probably say, yeah, the structure of that soil is pretty bad. Well, yeah, but we're able to put a number on it. And here, the sod version of the same, it's the same dirt, it's the identical twin that works out, or yeah, that has a healthy lifestyle versus the identical twin, the not so healthy lifestyle. We see what happens over here when we test this soil, this version of that soil, and everything's kind of in the green zone. Everything is good to go. Not just the nutrients and pH, but all these different parameters that we measure as indicators of health, as you'd expect under a sod. But if you notice this aggregate stability test, here's that 53% stable. That's 88. That's in the upper part of the green zone. That's really good. And if you look at this soil B, even from this little picture, you can see, oh yeah, that's got good structure. So what I'm demonstrating here is just that we can put numbers on these different parameters. Here's the soil structure one. That's easy to see. Some of these others, respiration, that's kind of hard to see or smell or kind of tell what's going on there. But you start to realize that soil A is probably going to have impaired respiration. It's probably going to have other issues besides just the structure, because it's remember, the balance between the soil health is a balance between these different features of the soil. And what this also shows is that most fields will end up being somewhere between A and B. So if you use this same soil in a more typical way, of course, it wouldn't look like soil A, and it wouldn't look like soil B. It'd be somewhere in the middle. But again, with the numbers, I could see, particularly if I'm measuring over time, which direction am I moving in? If one year I'm moving in one direction, or one rotation, or one cover crop, I'm moving in the direction of A, I'd say, well, that's the wrong cover crop, or maybe I shouldn't have plowed it. I should have left it on the surface and no-till. Same with, and that's just how the idea of using the information with a contrast. What I'm really trying to show here also is, if I know a good version of that soil on the right and a poor version of that soil on the left, I can learn a lot by comparing them. To understand the capacity of this soil, because it's only going to get so good. I'm probably not going to see on this particular soil aggregate stability higher than 50, then maybe 60 or 70%. Where some soils they're 100%. They laugh at all our drops of water, and they don't break down at all. Of course, they score 90s, high 90s or whatever. But that's okay. It's just, now I know I get a great deal of information from this contrast. That's what I'm trying to show you here. And so that was the first summary page. Second page talks about the scoring. Then pages 3 through 8 describe the different tests, how they relate to what the soil is supposed to be doing, and in respect to soil respiration or active carbon, and what your data show. Then on the last 2 pages of the report, it's a 10-page report, the general management practices that address this. Let me start then with that aggregate stability test, that one that was so low on the poorly performing soil, it didn't look so good. So this is the management suggestions table, and this is where we bring the lab data back to the field. And then we say in the short term, of course, this is just a starting point. But the idea is, if your soil structure is low, the idea is you could incorporate some fresh organic materials. Use shallow-rooted cover and rotation crops. We're not saying which ones, but we're just kind of setting a stage here where you can add manure. We call it brown manure. Green manure, you could grow a green manure. You could bring in mulch, depending who you are. That's kind of short term. And then long term, maybe reducing tillage, build up your soil structure a little bit, so keep a surface mulch. Rotate with sod crops that have maybe mycorrhizal hosts, again, to kind of build that soil population of creatures that will live and die, and of course, through their death, releasing glues and goos from their bodies as they cycle through, and helps glue the soil together. Another thing you notice here, we also talk about, for example, this fresh organic material, because all organic material is not created equal, right? The fresh material that's even identifiable yet, as that breaks down and goes through the soil food web, it's going to affect first the big animals, then the smaller animals, then finally the microorganisms. Where if you use a well composted material that you're incorporating, of course, you're missing out on the food opportunities for larger creatures, because they don't eat that stuff. It's already been eaten by someone else. And so you're feeding just microorganisms perhaps, and not the whole food web. So I mean, these are all things to keep in mind when we apply our management strategies to this information. And that's what I'm trying to say here. So the clever growers and consultants, they use this information just as a starting point to kind of work with the grower and come up with a plan. And that's what I show here. So here's that same soil on the right, the kind of beat up version of that soil. I show a grower and a consultant kind of looking over the results here, coming up with a soil management plan. So the way we do this, this is something we actually do when we have focus groups and stuff. We had field days and stuff when we'll all sit down and break into groups, and we'll give them reports like this, tell them the farm background, and then they'll kind of walk through this and talk about all these 6 kind of steps. And I think as anyone here probably knows, step 1 and 2 are oh so important. And the reason I am rather vague in general with our management suggestions table is because farms are different. Farmers have different goals. They're different people. They bring different things to the table. One person might be organic and use horses, and someone else might use giant machines. But you have to know that to understand any sort of planning or to be useful at all in discussing with the farmer, obviously, these results. So again, we start very vague in general with our management suggestions, but they're really agronomic principles to get you thinking about the principles, and then you want to apply them through the context of the grower. So I set step 2 here is to set goals and sample for soil health. Well, it looks like it's a little late if we're standing here with the report, but we'll get to that in a minute, how important sampling is, because this is an ongoing process. This isn't the first time or last time you're going to do this, but step 2 is just so important, and we'll see that why in just a minute. But so for each management unit, this is standard stuff. NRCS calls this something else, but it turns out we developed this, and then the NRCS has obviously a very similar set of steps to work with people, with growers, to just figure out what their constraints are, what functional groups are affected, what options they have for support, etc. But again, we want to talk about what's going on. So. And with this sample, you saw a picture of this beat up soil. We know it's got poor soil structures. I'm going to lead with that. So for number 3 here, identify constraints and prioritize. I'm going to say, gosh, we got to do something about that soil structure being so bad. But at the same time, I'm a clever guy. If I'm going to address the structure, the way I address that might be to get more organic matter in there, because that's low too. And I think we know by now that, yeah, by putting those two hammering our stone at those 2 birds, we might be able to bring both of those down, or in this case, bring both of those numbers up or down, as the case may be, but address both of those. And then here, our active carbon's low. How can if I'm going to add organic matter, maybe there's a type of organic matter that will provide extra active carbon. This is that fresh carbon, what we call sugar or candy for the soil microbes, as a type of carbon. It's easily oxidizable, that POX-C carbon. It's good to know this, because now I wouldn't want to add peat moss to this soil, because that wouldn't help me with my this candy, this sugar, this energy that I'm low in. So I want to grow a cover crop or whatever that would have lots of energy in it. And so I can start to put all these together, prioritizing it, and then say, well, what might I do? The management options, number 4, so important. If I said to the grower, oh yeah, just bring in some manure, he might say, well, the nearest manure is 40 miles away. There's no dairies around here. Well, I mean, I'd be looking for different work, right? But that's how important this is, and that's why we do start so general and leave it to the professionals and people that understand this, the soil health, and most people do by now, that you have to be very context sensitive of just how the different management ties back into these constraints which come from the measurements and what we're actually measuring. Of course, then this is to develop a short and long-term soil management plan. Again, that's just to get your head around all this information and kind of get it down on paper. And what am I going to do? Well, let's put some rubber to the road here. Sketch this out and say, what should I pay attention to? What should I look into? What should I pay? Right? What should I ask my cover crop seed salesman what he's got that'll take care of this and this? Because these guys do know. The cover crop suppliers they're very, very talented. Typically, certainly around here, very talented individuals that know what their cover crops can do. They've developed special varieties for this area, et cetera. And that's what I'm going to show here. So you talk a lot about cover crops, but as you know, the saying grow a cover crop, you're not helping anyone. And it's a good place to start. That's what my mom would say. Oh, is there something with cover crops? I read the newspaper. Well, no, of course, specialists know which cover crop and when, and which, as I say, which variety. And so you need to know, you don't just pick one that's easy to grow necessarily. That might be a reason, but you're going to want to try to address issues on that field or on your farm that are constraining, you've got a problem with. If you have a lot of erosion, well, you're going to look for a cover crop that establishes quickly, that certainly you have something growing during times of in the late fall or early spring. You want to have some green matter out there, of course. But so you take all this information together, and you're kind of holding all this in your RAM as a cover crop specialist. And then when you're thinking, yeah, what might we do to add a green manure to this field? If we're thinking that's the way to go. Of course, we want to be clever and put together our best package and talk to the grower and say, yeah, I'll find out if Tommy is selling a cover crop that can establish that late after your corn comes out. And as you know, now they've got more and more crops that are starting later and later. Seasons extending because of climate changing. So there's not just rye grasses that you can plant real late. They're starting to move. There's barleys that you can plant real late. It'll still establish before winter. All these different things. So there's all these different things you need to keep in mind, of course, when you put together this management plan. Because the whole purpose of spending this money and getting this information isn't to get a colored report that's all green or that's not a target. What you're after is understanding what's going on out there. And of course, there's the green matter, of course, that's so important. But it's also the management of that green matter with tillage. Tillage is important. Organic matter, as I said before, we need organic matter to decompose. It's food. That food, as that food decomposes, it releases all kinds of nutrients. Again, the living and especially the dying creatures of the soil food web, their bodies just break open, and out comes whatever their blood is. It could be goo, but that goo, glues together the soil crumbs. There's some other creature that moves in, starts eating that glue in a good way, and it's just a food web, and you need that to happen, and it will happen. But when you till, of course, you speed that up, and of course, it's a clever balance. You need to use a clever balance so that you don't overdo it and just over decompose your organic matter, because you need to have some of it stay and actually accumulate. So you need plenty of decomposition and accumulation. Sounds like you've got to eat more or add more. And that's the trick. So because when this accumulates, of course, that particularly in sandier soils, you know this, but your organic matter stores water very well, holds the soil open and loose to let water in. And then when it gets there, it helps store it, loosens the soil, leaves it open. Of course, you're storing carbon for all the right reasons, for all the different types of carbon there are, all the way from sugars to longer term carbon. Each type of carbon, as you categorize it, does something good. It's all good for the soil in this grand scheme of things. And the real trick is to manage tillage and cultivation carefully. Sometimes soils are sluggish, and it's worth running a disc over it very carefully, or in parts of it, or in areas, to kind of get things started, as they say, get some air into the ground. And that's all important. You have to recognize how to do that and when and how that might work. And the diverse kinds of organic matter. It's so important to vary organic matter sources, as you know, because the different types break down, feed different creatures, and the more varied and diverse of a body of creatures you have in the soil, the better off you are, the more resilience you'll have. Coming back to this, I put this slide in a second time. Again, I wanted to just emphasize all of this testing. This is so useful here to let me know, okay, what's going on in that field. Remember that this is that field that looked very very crusted. There's a lot going on here in the red zone, in the orange zone. This is not good. The yellow zone. You'd want things in the green zone. If they're not, there's something going on, that the soil's performing subpar, because par would be 50, right? A score of 50. And a bunch of these are below 50. So the soil for medium textured silo, it's performing below par. No surprise. But what might we do to put all these together and come up with a management plan? Next to last slide, I want to talk about soil sampling. So important. Remember, what you put in the bag is what's going to get analyzed. So that said, we're very keen on being wise and clever when you take a soil sample. So I'm going to start with a general soil sampling here up at the top. I'm not going to talk about the area that this is, and this isn't a whole field, but it's an area where I'm going to take one sample, and one sample that I'm going to submit is actually a bunch of subsamples. What I'm showing here at these circles, that's my 5-gallon pail that I'm walking around with. I'm kneeling down here, going to put a subsample, a 6-inch deep subsample from here in the pail. And I'm going to take 2 of those penetrometer measurements because I'm interested in the soil hardness. Then I'm going to move along, do it again. Take a subsample, put it in the pail, take two soil penetrometer readings, go across this area, and then I'm going to mix that up, take out, or we need 4 cups of soil. That's a quart of milk, a fat Ziploc bag. That's how much dirt we need. That would be our sample then. That's how we get a sample. But it's important to realize that this sample, if this was a 40-acre field, I would not recommend doing what you see here. I'd recommend going to a small area of it that you understand. If it's your field or a field you work with, you have to go to an area that you understand and want to know about or do know about and say, okay, this is very typical right here. I'm saying right above 1. I'm going to say this area right here is very typical for the whole field. So I'm going to sample here very well, and then I'm going to upscale the results to the rest of the field rather than blindly going out and sampling all this area and diluting what I know with this whole big field, because certainly there's more variability in this whole field than there is here. So I'd rather know what's going on here very well, and then I will upscale it and say, yeah, that's the same all across here. And then maybe it's different on the far east side here. Maybe I say, oh, the soil's very different here. Well, I'd say take a sample here for the west side and say, put that in one bag. And on the east side where things aren't growing so well, take a second sample and submit that here, sample B. So A's over here, B's over here, so that you can understand what is going on. Because if you sample from all this, include the good and the bad, you'll get some average that's nowhere. And you won't really learn anything. All you get is the average. And you want more than the average. You want to learn and you want to understand. And that's why I introduced this idea of, well, this troubleshooting. So here's a patch. It doesn't look so good. Area A growing well. This is conceptual, but you can scale this up, but it does fit in the picture well. And here's area B. So if I want to know what's going on in area B, I've done the same thing everywhere. Why isn't it what's going on in area B? Well, I might know it's a wet area or what have you, but you get the idea. What I do then is I'd go to area A and I'd take 5 subsamples and put that in a pail, mix it up. I'd call that sample A. And I do the same over here in sample B. I'd say, I want to know about this dirt that's right here. So I'm going to go just in this area and put those 5 subsamples in a pail, call it sample B. It's 2 samples, twice the price. I learned 10 times more than if I just took 1 sample, 20 times, 100 times more. So I'm going to target my soil sampling to get the representation of my questions. What's going on here? What's going on here? Maybe I know what I did here. Maybe I used a different tillage. Maybe I disced a second time here. Well, how did it affect the soil? It doesn't look like it affected the plants not well, but what did it do to the soil? Now I can measure and find out. So we call this contrast sampling, focused sampling, where you're deciding what you want to know, putting dirt that represents that in the bag, and then getting the results and following it up. Troubleshooting. Just really important. We talk about this all the time. It's just really the way forward. With a big suite of information like this, it's a shotgun approach. So you're going to use a shotgun. Have a good question, so that when things fall from the shotgun, when you see these red marks or orange marks that you weren't expecting, it can start to explain what you thought you saw visually or in your yields or what have you, and you can start to get information to tease that out. Last slide, I'm just to show you again who we are on the web. This is our submission form. We've got all sorts of information on sampling. It's got the submission form has our address on it. This is all what they call metadata, like who are you, what's your address, ag service provider, name of the sample, what testing do you want, GPS coordinates, crop information, what have you. I'm not showing page 2 here, it got too small. But on page 2, we have, for each of these rows, we got place for those penetrometer readings if you do them. Of course, all this information is in our soil health manual. God, this thing's 150 pages long, but it's all Well, all my slides are probably taken from in here or adapted from here somewhere. It's real straightforward, lots of colors, no complicated formulas, just real straightforward stuff. All the ideas are really well developed in real simple format, and it's a free download. That's the web address. So that's all I was going to present. And I guess we'll take questions, and I'll see what you guys think.
Yeah, thanks, Bob. Yeah, very informative presentation. I think, yeah, we'll jump right into questions. And Frank, I think I'll lean on you. I think you've been tracking some questions that have come in the chat. If you want to just jump on here and start sending some questions Bob's way, and we'll see how far we can get.
Awesome. Thanks, Tori. And yeah, thanks, Bob, for an awesome presentation. So we had a couple of questions in the chat that I want to handle first in the few minutes that we have. So looks like the first question we got was on, so it looks like it was based on page 1 of the soil test report. Those values do not have units assigned to them. They are just lab values.
Yep.
Wondered if you could speak to a little bit how those values were defined. What are those based on?
Yep. They're all, it's all on page 2. We didn't want it on the summary page. It just gets cumbersome. Most of them are percentages. Certainly all the carbon is in percent. The respiration is, for example, milligrams of carbon respired per day. Well, it doesn't mean anything to me, except the numbers. I guess you just have to believe from page 2 when it explains how we do the test real quickly, and what the numbers mean, what the ranges are. They just have to believe that since we're not showing the milligrams carbon per day, whatever, they just got to believe that this 312, or I can't quite say it, the 312 is in the red zone. And so it's it's all there. It's all very standard testing, available lots of different places. We're not the most different thing we have in our lab is this the rainfall simulator for the soil for the soil structure testing. And that's just something we've been doing a long time. And we really like the idea of seeing how soil crumbs that are dried and spread out thin hold up to raindrops at a controlled rate. But yes, and so again, that's a percent. So if we put 30 grams on here in the lab, we measure how much we got a little filter paper underneath there. So we put 30 grams of dry material here to start. Then when we're done, we lift this sieve off. Underneath there's this filter paper. We let that dry a little, put it in the oven overnight. The next day we weigh that filter paper. We started with 30 grams, and the next day I've got 10 grams of material in my filter paper. That means 1/3 of my soil failed, 2/3 was stable. So I've got 66% stability. So 2/3 of my soil stayed here at the end of the test. 1/3 of it broke through. And so it's just a percentage. And so all these numbers, they're real straightforward. There's no mumbo jumbo, as they say. It's all just what you'd think. It's all fairly linear. And it's explained on page 2, 3 through 8 in the soil health report. Every soil health report has it. It's a 10-page document. It's digital. You zip through it real quick and you can get whatever information you need and wonder what's going on with this respiration. What are the details on this? There's a whole lot in the report. And then you can go to the manual and look there and say, oh, I see. Because it really is straightforward information. It's stuff we just call it something that you have. Aggregate stability doesn't mean a lot to a lot of farmers, but crumb structure people do understand.
Gotcha. Thank you. Had another question. I think you covered some of this already, so I'll try and pick through. A big one would be like just the depth and type of soil sample. Are we talking like a probe or a shovel? And what depth are you wanting?
Right. I'll come to this one. Yeah, we're calibrated for 0 to 6 inches. That's just the way we've always done it. All our database is all scored on that. We could have done 8, but 6 capture. Then with 8, of course, it's much richer at the surface than even down right. Inch 4 to 5 or 4 to 6 isn't as rich typically as inch 1 to 2 going down, right? So it's important that with a soil probe, of course, when you take a soil probe, you get the same amount of material from every depth, right? And then you bring your soil probe up, and if you went to 7 inches deep, then you can, as you push the little core there into the tube, you can take that bottom inch, discard that, and take that 0 to 6 inch piece, dump that in your pail. We always recommend using a spade. What we do is we dig a little hole with the spade. You get very good with the spade. We dig a little hole with the spade, kick that to the right, just outside the hole. And then we come on the back side of that hole towards us, and we take what we call a slice of bread. We slice down 6, 8, 10 inches and bring that up sideways on the spade, trim that down right over the hole, so that what we have laying on the spade then is the same amount of, just like the soil core, the same amount of material from all the depths. So in 3 dimensions, I've got a bunch of soil on that spade that comes from 0 to 6 only. The deeper stuff, if it was on my spade, I kick that off the spade now. So as I'm standing there, I just knock that off back into the hole and put the material I want in a pail. That's my subsample. I move to the next spot. But so the idea is, yeah, 0 to 6 inches. We also recommend taking surface debris away. So if there's a huge amount of surface debris, I mean, it's literally you kick it with your boot, and just make sure that you don't have 2 inches of organic material that when you, like in the woods, then when you take your 6-inch deep sample, you've got 4 inches of soil and 2 inches of organic matter, unless that's what you want to know. If the researchers do all sorts of crazy things, but for a grower, we recommend that you discard it's a soil sample, 0 to 6 inches. So start with soil at 0, down to 6, no deeper, because if you have soil from 7 and 8 inches, that's much less even fertile than up above. You'll just be diluting your numbers, and the soil you won't get a true indicator value for the what's going on there in the 0 to 6.
Great, thanks. Had another question here, just asking what the status of a national soil health testing standard was. So if that was being in development, or what the status is of that.
Yeah, I think we're meeting today at 2 o'clock. 2 o'clock our time. We're having a meeting of soil health labs all over the country with the national, the national soil standards folks. That there's a real push to standardize the testing, so that, right, I'm saying we have a 4-day respiration. Other people are using a 1-day respiration. How do I compare these? All right. And so it would be nice to have just one way to measure respiration. However, as you know, the different even extractants for nutrients, there's a lot. We've wanted that for years, to just use one for the whole country. But the country is different. We've got different climates, different soils, different geologies, different weather. You really do. There's a great case to be made for really needing these different areas. Thank the land grants for this. They've really kind of done a great job of saying, for our area, we use a weaker, stronger solution to really get at what we need with the material we tend to have in this area. Midwest with Malik, or even Bray, as you go a little further west. There's reasons for these different chemicals, and there's good reasons for it. Then you try to calibrate one or translate one to the other, doesn't work so well. That can happen. So we get around that with our test. I said we use a modified Morgan solution, which is 10 times weaker than Malik. That's okay, because what we do, remember how we report it, we don't say how much to add. What we say is low, medium, and high. Low, medium. We give it a score, low, medium, high. And so if you're in the green zone, of course, doesn't matter what you extract. If we say it's green, I'm guessing if you extracted that with Malik, it'd also come out green. If we say, oh, it comes out yellow, let's say the phosphorus level was well, for us it'd be 8, and that would be yellow. Let's say it's 50 and it's yellow. I look over and say, oh, that's medium. So my phosphorus is medium. And a person might say, well, how much phosphorus do I add then? I'd say, I can't help you. You're medium. You should know now what medium means. I think growers have enough sophistication now to know that they're medium. They need to add phosphorus. And they know they're growing corn. They need 30 pounds a year to grow their corn crop. Corn is shipping 30 pounds of phosphorus off with every truckload that leaves with the acre of corn. There goes 30 pounds a piece. So they know that, and it's really just you're able to get you'll come out in the same range, and that's the information you need, as opposed to tell me how much to add. That's how we look at certainly the nutrients. And for the other parameters, it's the same thing. If our respiration test, our 4-day, comes out in what's our respiration test on this one, comes out in the light green zone, I'm guessing the 1-day respiration would also come out in the light green zone. It'd be a different number, but it'd be a different test. But I'm guessing it would also say medium or high medium or low medium sort of thing. So rather than getting hung up too much on the numbers, as important as the numbers are, certainly for researchers, the numbers are hugely important. But then, of course, in their research project, all they're on a field, they're all using the same test, so it doesn't matter what the test is, right? It's just they're contrasting A and B. It gets more cumbersome when you're doing big studies across the whole country. Again, people, instead of using the raw numbers then, because they can be so very different, factor of 10, they're using the scores and saying these management practices, this cover crop, based on the scores that happen when we use cover crop A and B. If the scores tend to be higher or lower, that's how we're saying this cover crop really affects respiration with the scores, because that universalizes everything and translates it into the common ground to compare test to test, place to place.
Great, thanks, Bob. I had a question, one here. If water management is a consideration for the management recommendations.
Only in a general way. We don't talk about, I mean, irrigation is outside my wheelhouse completely. However, I'm kind of a soil physicist, so I understand water holding capacity, how important water management is. And certainly here in the rain-fed Northeast, I can tell you that every drop of water that hits your field, wherever it hits, you want it to go in there. Person will say, oh no, my soil's too wet already. I wish it would run off. No one wants to run off for it typically. You're only going to get erosion. It ought to go in where it lands typically, because you're going to be wanting it later when it gets droughty. And so the idea is you want your soil built up and healthy enough, like a sod, even on a hill. So when the water hits, it doesn't end up in the ditch at the bottom of the hill. It goes in so that you've got it later. Now, if the soil's built up and healthy, of course, it doesn't end up being waterlogged and soggy. It kind of moves in and it percolates through. I said before that when the water comes in, of course, I like this one. As nature takes in a drink of water during a rain, she exhales the stale air that was in those pores and replaces it with fresh water. Then when that water moves down, she takes a fresh drink of air right behind the fresh breath of air behind that water that's percolating in. So the soil just gets fresh water and fresh air just by taking in that water rather than having it run off or being sealed. You get this water to perk through, kind of like a healthy sand. But of course, you want to move your soil into a condition more like sand with your aggregates. So imagine a well-structured clay, and there's no better soil in the world, because that well-structured clay, it takes in all that water and stores it in each of those little crumbs. But if it's well-structured, then there's all that water stored in there for later, but there's all that air as well, because the clay isn't breaking down into a brick. It's staying well-structured. I'm talking conceptually, but you get this idea that structure is everything, and getting the water in and down and storing it and moving it through, that's so important, because if you don't get the water in, let's say at the top of the hill, it's only going to overload the soil right below it, and the soil below that, and the soil below that. So if water's not going in at the top of the hill or the middle of the hill, the poor soil at the bottom of the hill is going to be over inundated with runoff. So it's going to get all this run on that it can't handle all that. And so that down at the bottom, or the stream or the creek or the road, you're going to have way too much water. So the idea is we want to control the water and use it and kind of keep it where it lands. That should just be the management target always. And the way to do that is build soil structure so that you can get it in and store it and use it later.
Thanks, Bob. I think I'm probably going to go ahead and kick it off to Helen. I have to apologize. We didn't get to all the questions in chat, but maybe there's a way we can share your contact information and have folks follow up directly with you. We can do that at a later time. But again, thanks for your presentation and willingness to answer questions. Helen?
Sure. Yep.
Yeah, good idea, Frank. Yeah, we can definitely share Bob's contacts so people can follow up afterwards. But yeah, we do want to run through a few quick other updates and reminders, and then we've got a QR code for CEU credits at the end here, so stick around for that if you're waiting for that. So just want to remind everybody we have one more session in our spring series on soil health tests. In April, we'll be joined by David Kleinschmidt, who is a consultant in Illinois, and he'll talk about how farmers should be using soil health test results to make management decisions on their farm. So if you're on this call, you're probably already registered for the series, but friends and colleagues can register on our website, ilsustainableag.org/ICCON. So hope to see you all next month. And then we also wanted to share the applications are open for ISAP's Soil Health Leadership Program. This is was previously titled the Advanced Soil Health Training. The content is the same and revamped, but yeah, we're excited to launch our 6th cohort of the training in August, and you can apply now through May 31st. It's an 18-month training program with 6 2-day trainings sort of spread throughout the 18 months. It's a free program, and like we're able to compensate meals and overnight hotel stays. The only thing we're asking participants to pay for is their travel to and from the sessions. So yeah, you can find more information about on our website. You can hear from past trainees. You can read recap blogs of what kind of trainings have we've included in the past. And yeah, you can apply on our website as well. So that's ilsustainableag.org/SHLP. And if you have any questions, feel free to reach out about that. And then just want to put a plug in for our cover crop t-shirt sales. Sales actually end today. They launched last month after ICCON call. But we worked with a local artist in southern Wisconsin to draw up these beautiful designs of cover crops, and we sell the t-shirts. The funding comes right back to ISAP to support our work. And it's a great conversation starter when you're out in the field. Great, yeah, for summer field days. They're in both short sleeve and long sleeve. So if you haven't gotten your t-shirt yet, we encourage you to buy one. And yeah, then just wanted to pause to see if there's other updates from people on the call. If people want to share events or other opportunities in the chat. And then we're also curious to hear from people, you know, after this series wraps up, we're always looking for what are the topics that people want to learn about. So feel free to drop some suggestions for that in the chat, but you can also send, throughout the month, send Rachel an email with any event that you'd like us to put on our website or share through our newsletter, and any topic or speaker that that you're interested in or that you've seen present that you think would be good for this call. We're always happy to take suggestions. So I'll just pause and see if there's anybody who wants to come off mute and share about an event that their organization is hosting or an upcoming workshop they'd like to share with the group. And if not, that's okay too. Yeah, you can sign up for our newsletter on our website and send out a monthly recap of upcoming events. So yeah, just encourage people to reach out to us with any updates they'd like to be included. And then I think our next slide is, QR code for CEUs. So if you're looking for CEU credits for attending, feel free to grab the QR code here. And if you don't have the CCA app, you can email Rachel with your CCA number, and we can list CEUs for attending today. And yeah, we'll leave this up for a second, make sure people have a chance to grab it. But I think that's all from us today. So thanks again, Bob, for joining us, and thanks for everybody for tuning in. And we're excited to see you all back in April.
Thanks, Helen. Thanks, everyone.